40,000 years ago, a group of humans crossed into Europe for the first time. They had never seen snow that didn't melt. They had never experienced a sun that disappeared for months. And every single one of them had skin that was almost entirely wrong for the environment they were walking into. For most of human history, skin color was not a fixed feature of any population. It was a slow, grinding biological negotiation between ultraviolet radiation, two critical vitamins, and a planet whose light keeps changing depending on where a species decides to walk. What separates the darkest skin on Earth from the lightest is not ancestry in the way people usually mean it. It is geography. It is latitude. It is a question of how much sun hits a patch of ground on an average Tuesday in December. And the full answer, the one that took scientists most of the 20th century to piece together, is stranger, more precise, and far more unsettling than almost anyone expects. The story begins not with color at all, but with fur. Around 8 million years ago, early members of the genus Homo were undergoing a transformation. The woodlands of equatorial Africa were giving way to open savanna. Hunting and gathering across long distances required hours of sustained movement under a direct tropical sun. Most mammals in that environment stayed cool by panting or limiting activity to dawn and dusk. But these early humans were doing something different. They were running in the midday heat, and they needed a cooling system that could keep pace. The solution was sweat, more specifically, a dramatic expansion of eccrine sweat glands across the entire body surface. But sweat glands work best on bare skin. So, gradually, the fur went. Nina Jablonski, a biological anthropologist at Pennsylvania State University, and the researcher who would eventually produce the first comprehensive theory of human skin color, identified the consequence of this transition clearly. Early hominins before the fur loss almost certainly had pale skin beneath their hair, much like chimpanzees, whose skin underneath their black coat is white. Losing the fur meant exposing that pale skin directly to the most intense ultraviolet radiation on the planet, equatorial Africa, where UV levels peak twice a year and remain punishing year-round. The genetic clock for what happened next was set by Alan Rogers of the University of Utah, who in a 2004 paper examined the human MC1R gene, one of the primary genes governing skin pigmentation. Rogers' team found that a specific gene variant responsible for producing rich, dark pigmentation originated approximately 2 million years ago. That date aligned almost exactly with the estimated timing of widespread fur loss. Dark skin, in other words, did not come first. It followed nakedness the way a shadow follows a body in sunlight. But here is the part the standard biology class version of this story leaves out entirely. For decades, the working assumption was that dark skin evolved to prevent sunburn. Later, skin cancer was offered as the explanation, the idea being that lighter-skinned early humans burned, developed melanoma, and died before they could reproduce. Jablonski investigated this theory in the early 1990s and found a serious problem with it. Skin cancers almost universally develop after the age of 40. In the ancestral environment, with a life expectancy a fraction of that, melanoma was never going to be a meaningful force of natural selection. It arrives too late to change who reproduces. The real answer came from a 1978 paper by two American medical researchers, Richard Branda and John Eaton. Their study, published in the journal Science, showed something that had gone largely unnoticed. Intense UV radiation doesn't just damage skin cells. It destroys folate, vitamin B9, in the blood. And folate deficiency is not a minor inconvenience. Folate is essential for DNA synthesis, for cell division, for the production of sperm, and above all, for the earliest stages of embryonic development. Without sufficient folate, neural tube defects follow. Spina bifida, anencephaly, miscarriage rates climb. Male fertility collapses. An hour of direct tropical sunlight is enough to cut folate levels in half for a person with no protective pigmentation. Jablonski recognized immediately what this meant. Dark skin wasn't primarily a shield against burns. It was a shield against reproductive failure. Every individual with more melanin in their skin was protecting their folate supply, which meant protecting their ability to have viable pregnancies, to produce healthy sperm, to pass genes on to the next generation at all. The evolutionary pressure wasn't slow and diffuse. It was immediate and total. By the time Homo sapiens emerged in Africa around 300,000 years ago, dark skin was effectively universal across the species. It was not one variation among many. It was the baseline. Now here's where the story turns. Around 60 to 70,000 years ago, a small population of humans, possibly just a few thousand individuals, began moving out of Africa. They moved northeast into the Arabian Peninsula, then into South Asia, then into East Asia, and eventually the Americas. Another branch pushed northwest into Central Asia and eventually into Europe. Every one of these migrations carried the same dark-skinned, folate, protected baseline into radically different environments. And in the environments farthest from the equator, something began to go wrong. Not with folate, with the other vitamin. Vitamin D is synthesized in human skin when ultraviolet B radiation penetrates the upper layers and triggers a photochemical reaction. In the deep tropics, with UV levels high year-round, even very dark skin allows enough UV through to produce adequate vitamin D. But at latitudes above roughly 50 degrees north, which includes most of Central and Northern Europe, UV levels in winter are so low that almost no skin-based vitamin D synthesis occurs at all, for months at a time. Without vitamin D, calcium absorption fails. Bones soften. Children born to vitamin D deficient mothers come into the world with weakened skeletons. Pelvic distortion from early childhood rickets was, for tens of thousands of years of human prehistory, a leading cause of death in childbirth, for both mother and infant. Researchers from Historic England and McMaster University, in a 2018 study examining 787 human skeletons from 18 cemeteries across the Roman Empire, found evidence of rickets in more than 1 in 20 children, with the highest rates appearing in northern England, where sunlight was weakest. That was just 2,000 years ago, when populations were already partially adapted to low-of environments. The selection pressure in the first millennia after humans arrived in northern Europe would have been orders of magnitude stronger. In that environment, the mathematics of survival quietly reversed. Dark skin, which had been a reproductive advantage for over 1 million years, became a liability. It was blocking the very ultraviolet radiation the body now desperately needed. Individuals with slightly less melanin, with mutations in genes like SLC24A5 and SLC45A2 that reduced pigmentation, could synthesize more vitamin D through the same weak winter light. Their children survived in greater numbers. Their bones held. But here is what almost nobody predicted. The assumption for most of the 20th century was that humans who arrived in Europe 40,000 years ago must have rapidly lightened to cope with the low-of environment. It seemed obvious. It seemed inevitable. It wasn't. In 2018, researchers from University College London and the Natural History Museum extracted and sequenced the genome of a Mesolithic individual known as Cheddar Man, Britain's oldest nearly complete human skeleton, found in 1903 in Gough's Cave in Somerset, England, and dated to approximately 10,000 years ago. The DNA was unusually well preserved. When the pigmentation analysis came back, the results stopped the team cold. Cheddar Man had dark brown to black skin, blue eyes, dark curly hair, and he had been living in Britain at roughly 51 degrees north latitude for his entire short life. He was not an anomaly. Dr. Tom Booth of the Natural History Museum confirmed that the Cheddar Man result was consistent with other Mesolithic remains across Europe. Hunter-gatherers from Spain, Luxembourg, and Hungary from the same period showed the same pattern. Dark skin, pale eyes. The genetics of sub-Saharan Africa in the middle of northern Europe, 10,000 years after humans first arrived on the continent. Then, in 2025, a team of researchers at the University of Ferrara, led by geneticist Guido Barbugiani and evolutionary biologist Silvia Girotto, published an analysis of 348 ancient individuals spanning 45,000 years of European prehistory. Their conclusion reordered the timeline entirely. Dark pigmentation was not just present in Mesolithic Europe. It was dominant. During the Paleolithic, the first 35,000 years of human presence in Europe, nearly all sampled individuals had dark skin. Even in the Copper and Iron Ages, roughly 5 to 3,000 years ago, half the analyzed individuals still showed dark or intermediate skin tones. The Barbugiani team put the date at which light skin became truly widespread in Europe at approximately 3,000 years ago, which is, to put it in scale, the era of the Trojan War, the founding of Rome. Humans had been living in northern Europe for 40,000 years before most of them turned pale. The process was not fast. It was not clean. It did not happen all at once. What explains the delay? Part of the answer is diet. Hunter-gatherers living in northern Europe before the agricultural revolution were eating substantial quantities of wild game, fish, and animal organs, all of which are naturally high in vitamin D. They may have been meeting much of their vitamin D requirement through food rather than through skin synthesis, which would have reduced the selection pressure for lighter pigmentation. When farming arrived in Europe from the Near East, beginning roughly 10,000 years ago and spreading across the continent over the following millennia, diets shifted dramatically toward cultivated grains and domesticated animals, which are far poorer sources of vitamin D. The vitamin D gap widened. The selection pressure for lighter skin suddenly intensified. A second part of the answer came with migration. The farmers who spread agriculture into Europe from Anatolia, beginning around 9,000 years ago, carried with them gene variants, particularly of SLC24A5, that reduced pigmentation. As they interbred with the darker-skinned indigenous hunter-gatherers, those gene variants began moving through European populations. A later migration of steppe pastoralists from Central Asia, beginning around 4,800 years ago, carried additional pigmentation, reducing variants, and pushed the process further. Light skin in Europe was not a single evolutionary event. It was the product of at least two major migration waves, depositing new gene variants into a population that was, for tens of thousands of years, still dark. The speed of the transition, once it finally came, was extraordinary. When the SLC24A5 variant swept through European populations, it did so faster than almost any other gene variant tracked in the human genome. The selective advantage was real, it was large, and it had been waiting, under population pressure, for the dietary shift that farming brought. And here, precisely, is the thing that should unsettle anyone who has spent time thinking about the social meaning of skin color. The variation in human skin color, from the deepest brown to the palest pink, represents, in total, roughly 2 million years of evolutionary history. The differences between the darkest and lightest human skin are not ancient, not permanent, and not predictive of anything except UV latitude. The MC1R gene variant responsible for darker equatorial pigmentation is one of the most evolutionarily constrained loci in the entire human genome, barely changed in those 2 million years, because it has always been essential. The variants that reduce pigmentation have appeared, spread, and changed repeatedly in different populations, through different genes, at different times. Light skin in Northern Europeans, light skin in East Asians, and light skin in certain South Asian populations did not arise from the same mutation. They arose through completely independent evolutionary events in different genetic pathways, all converging on the same biological solution to the same problem. Not enough ultraviolet B radiation to make vitamin D. The same environmental pressure produced the same result in separate populations through separate molecular mechanisms. Evolution, working on different raw material in different places, kept arriving at the same answer. Carles Laluisa Fox, of the University of Barcelona, who led the 2007 study extracting MC1R variants from two Neanderthal specimens, one from El Cidrón, Spain, and one from Monte Licini, Italy, found that Neanderthals had independently evolved their own pigmentation reducing mutation. Not the human variant, a completely different one, arising through a completely different molecular mechanism that produced a functionally similar result. Even the species that modern humans eventually replaced in Europe had already solved the Loewe problem through its own parallel genetic route. A 43,000-year-old Neanderthal from Spain, a 10,000-year-old hunter-gatherer from Somerset, England, with dark skin and blue eyes, a Bronze Age farmer in Hungary whose skin was finally, gradually beginning to lighten. All of them running the same calculation, generation by generation, with the same ultraviolet radiation, hitting the same square centimeters of exposed skin. The thing about skin color is that it appears, from the outside, to be a stable human category. Ancient, fixed, fundamental, the actual biology tells a different story. In 2000, when Nina Jablonski and George Chaplin published their landmark paper in the Journal of Human Evolution, the one that used NASA satellite data on global UV radiation levels to map predicted skin color against the actual skin colors of indigenous peoples around the world, and found an overwhelming degree of correlation. Jablonski described what the data revealed as a beautiful sepia rainbow, not discrete categories. A gradient, a continuous biological response to a gradient of light. The humans who first crossed into Europe 40,000 years ago had skin that was, for the environment they were entering, entirely wrong. But wrongness in biology is not permanent. It is just a problem that is waiting for enough cold, dim winters, too.